So hello guys and welcome back to another very important video session from your own channel Let's Crack NEET UG English Breaking All Barriers, a very good initiative by Unacademy. So I'm your friend Vipin Sharma and your biology mentor as well. In this particular video we are going to revise a very critical topic from examinations point of view for NEET 2020 aspirants, that is the lactose operon, which you commonly know as a smaller name called as LAC Operon. So let's discuss this topic and let's crack it guys. So these are some important credentials of mine. You can have a look if you want. Otherwise, let's proceed. You can also get connected to us via Unacademy's learning app. This app is freely available on Google Play Store. You just have to install this app, and after that you just need to go to the NEET UG section, and after that you need to click on the get subscription icon. When you are going to click on the get subscription icon, you will be getting all our subscription packages on your screen, starting from the duration of 1 month to a duration of 24 months, that is 2 years. But I will surely be guiding you in a proper way if you will take a subscription of 1 year and 2 years, because it is a proper time to crack this entrance examination. You know very well that the preparation of this exam can't be completed in 1 month or in 3 months or in 6 months. So the ideal plan or the ideal syllabus completion could be done by you and by us also in a proper time of 1 year and 2 years only. So please make sure to enroll in our batches. You have to take a subscription of 1 and 2 years because these are financially very feasible batches. Let me give you an example. Let's say you are going to take a subscription of 1 month. You will be getting it for 5,400 rupees per month. So your monthly cost is 5,400, but if you will watch the cost of this pack of 2 years, then the monthly cost is only 1,300 rupees. So from 5,400 rupees to 1,300 rupees, it has reduced to 4 to 5 times. So it becomes very important for you to understand the importance of more bigger pack. Other than that, if you want to get more discount, then you can use this code Vipin. You will be asked a referral code before paying your subscription fees. So if you will be getting your subscription by utilizing the code Vipin, then you will be getting 10% instant discount, and after that you can watch all the lectures of all 40 plus educators on Unacademy Plus platform. This code is most important for you guys, and you will be directly entrained in my personal batch, okay, on Plus. So I'll be completing your biology syllabus, and other than me you can watch the videos of all different educators on Unacademy's Plus platform. Plus you will be getting all these kind of perks. You will be getting live classes on daily basis. You will be getting structured courses. It means that no random videos will be given to you. You will be guided in a proper way. There will be live tests and quizzes with live polls. Plus you will be getting ranks on daily basis. It means that if you want to improve your rank, then you can track your ranks on daily basis, that on day 1 that you are on, let's say, 100 rank. After 30 days you are on 70 rank. After 30 days you are on 10th rank. After some weeks you are on 2nd rank. So you can just track your performance according to daily poll analysis and daily rank observation. Plus you have the unlimited access. Let's say you have a 2 year of subscription pack, then for these 2 years you can watch as many videos as possible, you can watch as many live lectures as possible, and you can follow as many teachers as possible. That totally depends upon you. You will be given a proper authentified course in this particular subscription. So please make sure to use the code Vipin. It is really gonna be helpful for you guys because you will be getting the whole syllabus by applying this single code only. Now let's talk about the LAC Operon. And now let's talk about the basic components of the LAC Operon. Firstly, you need to understand that what an operon is, okay, and what are the different kind of things that makes an operon. There are 2 different kind of organisms. You know about them very well. The first type of organism is called as prokaryotic organism. So what are prokaryotic organisms? Prokaryotic organisms are those organisms that doesn't have a well-defined nucleus. Other than that, there are eukaryotic organisms. So eukaryotic organisms are those organisms that do have a well-defined nucleus, okay. So in those prokaryotes, the arrangement is called as polycistronic, and in eukaryotes the arrangement is called as monocistronic. Now let's just have a look that what this particular arrangement is. P for prokaryote and P for polycistronic. So you do not need to get confused between these 2 statements. The polycistronic word is just for the prokaryotes, and the other cases, that is monocistronic, is going to be for eukaryotes. There should be no confusion in this. Now what is this polycistronic and now what is this monocistronic? This thing need to be understood by you in a great way. Polycistronic means more than one cistron. Okay, cistron means the coding part. So more than one cistron is getting expressed by a single promoter. So a single promoter, a single operator is basically guiding the transcription and translation of more than one cistron, then it is called as polycistronic. But in case of eukaryotes, it is monocistronic arrangement. It means that a single cistron is only expressed by a single promoter and operator. Now let's understand this case in a better way. Let's say that this is the part of a DNA. And let's say there are different cistrons. Let's say this is first cistron. Okay, let's call it C. This is second cistron. Let's call it B. There is another cistron. Let's call it A. And let's say these three genetic fragments code for different kind of proteins. If these are part of DNA, then we know very well that this DNA will be converted into RNA, and that RNA would be converted into a protein. That is very obvious thing that we already know. Now what you have to do is, there is only a single promoter and single operator which is present over here, right? Now this single promoter would be guiding the expression of A, B and C. It means that if this promoter is activated and if the transcription is getting started, then transcription of all these three cistrons, that is A, B and C, will take place, and they will hence be expressed by the effect of single promoter and single operator, right? So this is the particular thing that you have to understand in polycistronic arrangement. Now what happens in the case of monocistronic gene? In case of monocistronic gene, it is quite different. Mono means one and cistrons mean the genetic fragment that is going to code for a polypeptide. So if this is a particular DNA sequence, let's say this is a cistron A and this is a cistron B, then it will be having its separate promoter. It means that if you want to understand it in terms of a bulb, then what happens in the case of eukaryote? Then in case of eukaryote, it is a bulb that have a single switch. It means that this bulb has a single switch. If you will be keeping this switch on, this bulb will be enlightened, and if you will be keeping this switch off, the bulb will not be enlightened. So only a single switch is taking care of a single bulb. But what is the polycistronic arrangement? It is the a series of lights, as you used to use at your homes at the time of Diwali. There is a series of lights, okay? There is a combination of lights having only a single switch. It means that if you are going to keep this switch on, all the lights would be enlightened at the very same time. It means that they all will be expressed just by the same promoter. But here each and every light, each and every separate bulb require the special kind of promoters to be expressed first, so that they could be enlightened. So this is the difference between monocistronic and your polycistronic genes. After that, you need to understand the importance of the split gene arrangement. So the gene arrangement in case of prokaryote is very regular. Okay, let's say in case of prokaryote, it is the whole gene. There are no introns, there are only exons. So there is no split gene arrangement, right? So what happens over here is that if this is the 5' segment, if this is the 3' segment, this is the whole genetic fragment. These all are the coding segments called as exons. And since there are no introns, then you don't call it exons or introns, you basically call it the coding segment. But in case of eukaryote, there is split gene arrangement. Split gene arrangement means between the coding fragments, there are non-coding fragments which are present. Let's say that the blue ones are coding fragments. Okay, this is a coding gene, this is a coding gene, this is a coding gene. Let's say that these are basically coding in nature. So what happens over here is that this is the non-coding gene, This is the non-coding gene or basically the non-coding segment specifically, not the gene. This is also the non-coding segment. So what is happening over here is that all these positions are basically coding in nature. So there is no split gene arrangement in case of prokaryote, but in case of eukaryote there is a coding part called as exon, then there is a non-coding part called as intron, then there is a coding part called as exon, then an intron, then an exon, then an intron, starting from 5' to 3' position. It is called as split gene arrangement. Split gene arrangement, which is a characteristic feature of your eukaryotes, but it is not found in case of prokaryotic organisms, right? Alright. So what are the different parts of an operon? Let's talk about that. So different parts of an operon could be easily memorized by a very simple trick that is called as R-SOAP. Okay, R-SOAP. So let's make it quite beautiful so that you can understand each and everything in a better way. So it is R-SOAP. Now what are these R-SOAPs? Let's talk about that. Okay, what are the full forms of these R-SOAPs? So R is basically what? It is a regulator gene. It is a regulator gene. S is what? It is the structural gene. It is the structural gene. O is what? O is basically the operator. And P is what? P is basically the promoter. Okay, so there are some things that you need to understand. P for promoter, and P is the place where RNA polymerase used to bind. So P for promoter and P for polymerase. So polymerase used to bind at this particular position. O is operator. Okay, so here the repressor will bind. The repressor will bind at this particular place. Structural gene will code for the proteins, or the mRNA. It means that these are the structural genes or the DNA fragments that will be converted into RNA fragments, and then those RNA fragments will be directly converted into a protein. So that is done with the help of structural genes. And then there is regulator, which helps in the regulation of operon. So now let's come to the topic. What is the topic? The topic is LAC Operon. And in LAC, what LAC basically stands for? In LAC Operon, the LAC basically stands for, the lactose, which is a kind of sugar. So in LAC Operon, the LAC word basically stands for the sugar, that is lactose. So in LAC Operon, there are 3 different structural genes which are encoded. So what are these 3 structural genes? These structural genes are gene Z, gene Y and gene A. So what is Z? Z basically codes for beta galactosidase. Y basically codes for permease protein or permease enzyme, and A basically codes for trans-acetylase enzyme. So now what are the important functions of all these 3 enzymes? We need to ponder on that. So basically beta galactosidase, as the name indicates, it will act on the beta position, and it is galactosidase. So it will be converting the lactose into glucose plus galactose. It will break down this disaccharide into 2 monosaccharide units. That is very, very important. What is permease? The permease will be increasing the permeability of the lactose. It means that if the lactose is present around the bacteria, let's say this is the bacteria, and if the lactose is present in the surrounding, then that lactose will be coming via a particular path, and that particular protein that will let the lactose come inside the bacteria is called as permease. It means that if there is minimum amount of permease, then only your lactose can come inside the bacterium, which simply let us know a fact that when there is no permease, there could be no lactose inside the bacterial cell. So if you want the lactose to come inside the bacterial cell, you want this permease to be expressed at least in a small amount. At least in a small amount. Because when the permease is getting expressed in small amount, it will let the lactose come in, and when the lactose will come inside the bacteria, the beta galactosidase will break down that lactose into glucose and galactose, which will be directly utilized as a source of energy. Trans-acetylase. So at the trans position, acetylase simply means the enzyme that incorporates the acetyl group or basically remove the acetyl group from a particular enzyme. It means that acetyl acts as a switch. So if there is addition of acetyl or removal of acetyl, it will make a protein go on and it will make a protein go off. It means that it will either turn it on or it will either turn it off. So that is the work of your trans-acetylase. So trans-acetylase will be regulating the function of Z and Y. It means that all these three enzymes are having the function of metabolism of lactose only. So if there is no lactose, none of the enzymes will be expressed, but a minute level of expression is needed for permease so that the lactose could come inside the bacterial cell. So in the absence of lactose, all these enzymes will be turned off. But when the lactose will come, the permease will let the lactose come inside the bacterial cell. The beta galactosidase will be converting it into monosaccharide, and the trans-acetylase will be complementing their function by the acetylation deacetylation process. Now let's talk about the diagram that is given in NCERT. So I have given you enough information to understand this particular topic in a better way. So let's talk about the sequence of genes. What we have understood till now, that there is PIPO, ZYA. So here in this bacteria, this is a polycystonic arrangement. How? Because this is polycystonic because this single promoter will be activating the Z, the Y and the A also. So single promoter is helping in the expression of Z, Y and A at the same time. So it is called as polycystonic. So let's say the inducer is absent. What is inducer? It is lactose. So let's say there is no sugar in the atmosphere. So will the bacteria be expressing Z, Y and A? Obviously not. If there is no lactose, then there is no need to break the lactose. What are the functions of Z, Y and A? Beta galactosidase, trans-acetylase and permease are having the ability to lyse the lactose or to break down the lactose. If there is no lactose, if the lactose is absent, then why would they be doing this kind of function? Obviously they won't be doing that, right? So in the absence of lactose, Z, Y and A will not be produced. And why they are not produced? Because there is something called as a repressor that stops the expression of Z, Y and A. So what does the repressor do? The I gene codes for a repressor, and repressor protein which is activated, it goes to the operator, it binds to the operator. So there is an important point you need to note down, that repressor binds to operator, right? Repressor binds to the operator. So repressor and it goes and binds to the operator. Now let's say that the polymerase is also present at the promoter. Will it be going forward? Obviously not. Let's say there is a polymerase which is present over here. Will it be able to go in this particular direction? How does the polymerase work? The polymerase need to go from this particular direction to this particular direction so that it could scan the Z, Y and A and it could express them. But what is happening over here? In this particular case, the polymerase can't go further because there is a big hurdle, there is a big rock in the path. And that rock is what? That rock is basically a repressor which has binded itself to the operator. Okay, so there will be no transcription or no translation in this particular case. So at the end of the day, you will be getting your products in the form, you will be getting no products over here because these are not getting expressed. But let's say your inducer is present. Let's say your lactose is present. So what does the lactose do if it is present? The lactose used to inactivate the repressor. The repressor which was earlier binding to the operator, now it will not be able to bind. Why? Because it is inactivated by the inducer. So when the lactose or the inducer bind, to the repressor, it used to inactivate it. And this inactivated repressor can't go further to this operator. So the operator is now free. There is no stone in the path. So what will happen? The polymerase which is present at the promoter region, it will go in this particular direction freely and it will scan Z, Y and A. When it is going to scan Z, it will be forming an complete mRNA and these all cistrons will be expressed. The Z will be forming beta galactosidase, the Y will be forming permease and the A will be forming trans-acetylase over here after translation. So if the lactose is present, then the enzymes which are able to break the lactose will also be activated. Why? They will be activated because lactose acts as an inducer and that inducer used to suppress the activity of the repressor. But when the lactose is not present in the atmosphere, then the bacteria will be just expressing the repressor which will bind to the operator, which will not let the promoter to go to the Z, Y and A regions, and ultimately there will be no transcription or translation taking place. So this is your LAC Operon and it is called as an inducible operon. It is called as an inducible operon. Why? Because you need to induce that. You need to induce that means you need to keep it on. You need to make it on. You need to turn it on. It means that in normal situation it is off. Normally it is off. You need to turn it on. You need to turn it on. So if there is no lactose, then this particular system will be at the rest. This particular system will not be expressed. It will be in the off state. But when there is some sort of lactose, then this system is going to express itself. The Z, Y, A will be expressing themselves and at the end of the day you will find this system turned on just because of the inducer, that is lactose, because it will suppress the activity of repressor that will be going to the operator and hence the promoter or the polymerase binded to the promoter will not go further. So this is very, very important from examinations point of view. Thank you so much guys for watching this video up. If you really enjoyed this session, then please make sure to hit the like button on this video. You can also subscribe our beautiful channel because this channel is really going to help you out for NEET 2021 and NEET 2022. And there are also a lot of videos coming up for NEET 2020 entrance examination so that you can revise your concepts in a better way. I used to take a lot of special classes for free on my, on Unacademy's learning app. So you need to install that app and you need to search for my user ID, that is Vipin 007, or my name, that is Vipin Kumar Sharma. You can directly follow me up over there so that you can never skip any of the session from my end. Other than that, if you want to get your syllabus completed by me and rest of the 40 top educators of Unacademy, then you can use this code Vipin. You will be getting 10% instant discount on your subscription and hence you will be completing your syllabus in a better way from the top educators of India in the best batch of India, and you will be qualifying your examination very easily. That is a sure bet from my end. So please make sure to use this code Vipin so that you can enroll yourself up into my batch and you can complete your syllabus in a dramatic way with a lot of fun, with a lot of knowledge. Thank you so much guys, lots of love. I'll surely catch you in the next video. Thank you so much. Thank you so much for supporting me so well. Bye.